Hi, today we're going to talk about codon charts, and not just what they are, but how to read them as well. You're going to see a lot of familiar vocabulary, and we're going to make sure that we clarify so that there's no misconceptions as we continue on with this information. So first, let's start. What are codons? Well, codons are three-letter sequences of nitrogen bases on messenger RNA. Each group of three letters, each triplet, tells a cell which amino acid to add next when building a protein, or when to start or stop making the protein. So you have codons that tell your cell this is the start and this is the end of your amino acid chain, and that amino acid chain will determine the shape of a protein, and the shape determines the functionality of a protein. So we've seen a lot of examples of codons, and they're, again, a triplet, so they're always going to be three. So AUG, GCU, UAA. These are just sets of three along that messenger RNA, and we read them in codons, in triplets, because that's how your transfer RNA is going to bring the specific amino acid to the ribosome. So before we move on, we need to make sure we really know this. Each sequence of three nucleotides is the codon. That's what we're referring to. The genetic code is just a set of instructions that translates the genetic material into proteins. And remember, proteins are a biomolecule that can be so many different things, from enzymes to structure. This code defines how sequences of three nucleotides specify which amino acid will be added next during protein synthesis. And don't forget, protein synthesis isn't something to be intimidated by. It's just making proteins. So what's the importance of a codon chart? Well, in my opinion, I would rather not have to memorize all of this. So a codon chart acts like a decoder tool. Think like your 1940s spy, and you need to figure out which amino acid each messenger RNA codon stands for. That's the purpose of a codon chart. What do scientists and students use a codon chart for? Well, they're going to use it to translate messenger RNA into the correct sequence of amino acids. That sequence of amino acids determines the shape and function of any protein. So you need to get the shape right, or the protein's functionality won't be right. And we talked a little bit about this when we talked about mutations, like sickle cell anemia. The shape of the protein had a mutation, and it didn't code correctly, which is why you end up with, instead of soft, flexible blood cells that can carry oxygen very efficiently, you end up with rigid C or sickle-shaped blood cells that don't carry oxygen as efficiently. And it's because something went wrong with the shape of the protein. And that problem happened as a mutation along the codons. So how do we read a codon chart? Well, there's two types of codon charts that are the most common, and we're going to talk about both of them. I have a preference, and it's going to be the first one we talk about. It's the circle codon chart. This is my favorite. I think it's the easiest one to work with, and you might disagree, and that's okay. That's why we'll talk about both, because you're going to see both. So let's code for GGG. Nice, easy, straightforward. So first, for a circle codon chart, you start in the middle, where you have G, U, A, C. So to our first letter of the codon, GGG, is G. So we find G in the middle. Next, you're going to find the second letter of the codon, and that's also G. So you move around the circle. So you start in the middle and you work your way out. Then we find our third G, and again, we work out. And it tells us what we're looking for. And here we see that it's glycine. So the codon G, G, G codes for glycine. And we figure that out by starting in the middle and working our way out. So circle codon charts, you read from the inside to the outside. And I think it's just very straightforward and natural to do. And you get glycine at the end of G, G, G. This is the other type of codon chart. I think it's less user-friendly, but it's not hard. So let's code for the same codon, G, 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 and we already know it's going to code for glycine, but that's fine. We are reading the chart. We don't need to rework the wheel every time. So first you find your first letter, and we know it's the first letter because it's going to be in the first letter category. So these are all representing the first letter. Next, you're going to make sure that you're only looking in the G section. So it goes straight across this rectangular codon chart. Next, we're looking for the second letter, the second G, and we find it right here. And then we've narrowed it down to these codons within the first and second letter. And remember, we've talked about how codons have a bit of redundancy built in. And that's how we get silent mutations. And a silent mutation can happen if you have a substitution within your codon. So one of your letters, one of your nitrogen bases gets switched. It's not correct. But you have a silent mutation because no one's going to yell, help, because it's a silent mutation, because no one's going to notice. So you could have G, G, U, G, G, C, G, G, A, or G, 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 and it would code for the exact same amino acid. This is just one of those silent mutation possibilities, where you've had a mutation happen and a substitution occurred within your codon, but it accidentally, coincidentally, coded the exact same amino acid you needed. So again, we're practicing reading the codon chart. So we have to find that third letter G, and we find the third letter, it will be along this last edge. So let's find out. So again, you start with your first letter, you find your second letter, and then we need to find our third letter, and it's another G. So G, 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 and it's going to be glycine. So we've looked at two different ways of reading we've looked at two different codon charts that are read very differently, but we found the same answer. We found the same and accurate amino acid. I think the redundancy is a lot easier to see on the rectangular codon chart, but I think reading the circular codon chart is a lot easier. So hopefully you feel a lot more confident going into more codon chart reading, and I can't wait to talk about more codon charts and mutations before we get into our next unit on meiosis. So have a great day. Bye-bye.